9 a.m. after the very fun seminar dinner last night. So I want to thank, so I'd like to thank the organizers. Actually, I'm listed as a co-organizer, but Sam did a lot of the work, and so I want to thank Sam for involving me in this, and all the other speakers. I really like coming to this conference, and this community, which I feel like is I'm sort of tangential to, but really enjoy interacting with you guys. So I'm going to talk about work that, so this is unusual in that I'm going to talk about work by two other PIs. This is all in collaboration with Omer Dushek, who's here in the UK at Oxford, and actually Sam. Jesse Goyette is a former postdoc in Omer's lab who did a lot of the experiments that I'll show, and now he's moved on. He's now a group leader at University of New South Wales. All these people sort of drove this project, and I feel like Omer and Sam are sort of at the mentoring level of what I did here. So I'll give you the broad picture of the system that we wish that we're trying to study. And the system that we're actually studying is simplified this, but all this is happening in the bio background. So this is my group. We're at UC Irvine, one of our main threads is that we're interested in the cell surface, so the eukaryotic cell surface, and mechanics at that surface, so how it experiences force and how it produces force. And the cell surface is a really interesting place. We became interested in this because of transient cell-cell contacts. And so these two red lines are supposed to be zoomed in, two cells coming together, as they often do in immune response, or other times during development, where cells will transiently come together, and then after some period of time, seconds, could be days, will leave. And one of the research projects that I'm doing with John Lowengrub and collaborators is to understand the role of this thin layer of water, this sort of 100 nanometer layer of water flowing back and forth in between the two cells. And we think that the water is doing something biologically significant. Then there are also large-scale deformations. So this is a micrometer scale simulation where there is a deformation of the cell surface. This is a cellular bleb, which is a hydrostatic pressure-driven deformation. And sometimes blebs travel, which means there's traveling wave phenomena, and so we study those too. So the cell membrane can deform. And then, because the cell surface is enclosed by a plasma membrane that is fluid, it's a two-dimensional fluid, that means many molecules, not all, but many are mobile, and so they undergo lateral diffusion. They move around laterally on this two-dimensional surface. And so for a long time, we wanted to understand the behavior of those. Sometimes they interact approximately, are independent sometimes. And we were able to make progress on this problem with Jay Newby. And actually, this project with Jay started at a conference a year and a half ago that is in some ways like the predecessor to this conference. So I'm really excited with that. And actually, I think Jay was here, and he talked about this project a few months ago. Right, so all of this is happening at the cell surface, and that makes everything very complicated. And we typically choose projects that focus on one of these aspects. And so all of this is happening, but basically what I'll talk about, we're pushing all this to the background. And the motivation for this talk is shown in this cartoon. So this is supposed to be a zoom in, just a caricature of the cell surface. This is the outside, and this is the inside, and these are molecules of the cell surface. This is one of the immune cells called an NK cell, which is a natural killer cell. It doesn't so much matter what the identity of these molecules are. They're receptors. But the point is that they're tethered to the surface, so they're sort of tied to the surface, they're stuck there, and there's multiple molecules, and they're interacting with each other, and they're interacting with molecules that are in the cytoplasm that are down here, and those ones are free to diffuse. Oh, and there's one called SHIP1 that I'll talk about. So this is not unique to NK cells and to these receptors. If you sort of dig through the literature on any type of cell signaling, which is how a cell interprets information from the outside world and eventually makes a decision, then you always get cartoons that look like this. So this is another one by about the T cell. This is the T cell receptor, sort of the most famous receptor here on the surface. And then this whole orchestra of intracellular molecules. Other receptors, there's something called PD-1, and that's gaining notoriety right now because it's a target for cancer immunotherapy. And so if you target this PD-1 receptor with certain drugs, then you endow the T cells with an enhanced ability to fight off cancers. And all these molecules that are tethered, interact with molecules that are downstream. Here's SHIP1 again, SHP1, and that's immediately downstream of PD-1, and then a whole bunch of other molecules. So this cartoon, everybody draws it in a different way, but the story's all the same. You have these molecules that are tethered, and then you have other stuff that's going on inside, and it can get quite complicated, as in most of these signal transduction networks. There's some complicated cascade. But there's ship 1 again, so I guess it keeps popping up. Here's a further artist rendition, same thing. You've got all these molecules that are tethered, and then so on. And so this is background fact number 1 that I want you to take from this, is that a lot of signaling molecules are tethered to the surface. And a lot of the signaling has to be happening at the cell surface, because that's where you import information from the outside. Okay, so then fact number 2 is that many of these molecules, maybe almost all of them, form clusters where between 10 and 100 of them group together after they're activated. So, so Dan Coombs talked about this on Monday, and you see this all over the place. So this is a signaling molecule called LAT. It's monomeric. They exist alone, and they're diffusing in 2 dimensions on the outer membrane. Then after something happens that triggers a signal, and then they bunch up together. And most of these clusters undergo some kind of maturation process where they become sort of even tighter. And if you're the type of person who's interested in the actin cytoskeleton, F-actin is implicated in almost all of these receptor clustering phenomena, in the sense that when you perturb actin, you start perturbing the clusters, and the mechanism is not really clear in many cases. So a lot of these receptors form clusters. Okay? And so a lot of people want to know why, in the sense of what is the mechanism that drives cluster formation. And so there's one group, just from a few months ago, this is Ron Vale, John Ditlev, and collaborators. They made an in vitro reconstitution where the molecules were mobile. This is actually LAT. They do something that sort of activates the signal, and then the monomeric transforms to clusters over a timescale of minutes. And then they add something else that deactivates the molecule, and then it goes back to being just a homogeneous cloud. Okay, so a lot of them form clusters, not just LAT. And all this raises this question, which is, what are the differences in terms of chemical kinetics, internal kinetics, and ultimately in terms of signaling between when they're not clustered and when these tethered molecules are clustered? Okay, and that's really the question that's driving us. We want to understand what is it that's happening in this cluster. And so just to be clear, there's really 2 questions. One is, what drives them together? Okay, and the experimental picture I showed you on the last slide is part of the answer to that, although it's still not completely clear. And then once they're within a cluster, so once they're all clumped together in this densely packed environment, what is special about that place that makes it different from the other places? And like in all biology, there's this sort of evolutionary design question, which is why do cells find it useful to make clusters? And we see it recurring over all of these signaling molecules. Okay, and in every case, they're tethered, and by tethered, what we mean is in the cartoons, there's always a little line connecting it to the cell surface. It's either a squiggly line or it's a straight line like here. So there's some mechanism that keeps it at that surface. Okay, so that's our question. I'll tell you about this experimental setup that Omer built. And like I said, he's really the driving force. Basically, the rest of the talk is us trying to make simulations to understand what Omer did. And then I'll go through sort of sequentially our modeling efforts, and Sam is involved in this, especially at the beginning, and then I'll take on another direction while we're trying to build further methods and eventually combine them. Okay, so here's the experimental assay. This is Omer's new invention here, and so just by way of comparison, this is sort of what you would do in classic enzymology. If you want to understand ship 1, so here's ship 1. It's one molecule here. In cartoon, it's drawn as 3 different domains that are tied together by these little linkers, but it is just one molecule, and it interacts with something that in situ is tethered, but in classic enzymology you untether it, and then you just mix these together, and they react. And you can either think of it as a well-mixed system, or you could think of it as a reaction-diffusion system that might form spatial patterns. But in either case, both molecules are free to move, so either by diffusion or you just assume well-mixed. We want to understand tethering at the cell surface, and so Omer's assay is to take a surface, tether them using an artificial linker, and then the actual protein of study, something called layer, that's tied to the surface here, so that the anchor point is immobilized. So the anchor cannot move laterally. And so this is reminiscent of what you have if things were really close packed and you can't just move. And so what that means is that one of these molecules has some kind of range, shown by this dashed line, especially if it's tethered to something a little longer, it can only reach some of its neighbors, and it can't reach further, and it never will, because it's completely immobilized. And then Omer flows in the cytoplasmic molecule of interest. In this case, it's SHIP, and then a whole bunch of chemistry happens. And so you look at this picture, and it looks different than the clustered pictures in a few key ways. One is it's infinite in extent, or approximately infinite in extent, whereas clusters are finite, but clusters are still pretty large. And the other one is that it's probably not as densely packed as what they are in a cluster, but they're still much more packed than they would be in solution, and it allows us to access some of what we're seeing. It's more reminiscent of a cluster. And that's the trade-off is slightly less realistic, but we can get high-quality quantitative information. And so then once he's built the system, he uses something called SPR, surface plasmon resonance, where you flow in the molecules, and then you get to watch how much is bound, how much SHIP1 is bound over time. Okay, so that's the setup. Here are the players. Here are the molecular participants. There's just two. So there's SHIP1, which is cytoplasmic. It's floating around in solution. It can do two things, so I made two little dots on it. Then there's the surface tethered molecule, which we're studying, is layer. And like I said, it's attached to the surface with a linker that is tethering it there, and the linker has a certain length we'll call L. And it comes in two states, either phosphorylated or dephosphorylated. And in our system, once it's dephosphorylated, it doesn't really do anything. So you can think of that as like a null state. But they start off all phosphorylated. You can think of it as active. SHIP1 can bind to phosphorylated layer, and once it's bound, it can dephosphorylate neighboring layers. And so that's all shown here. So A is the tethered molecule, B is SHIP1. They can reversibly bind, so there's a K on and a K off. And then, once you have this complex, it can kill an A. Right? It can dephosphorylate one of these A molecules. And that happens at some rate, Kcat, for catalysis. Okay, so this, if you start sort of forward simulating this in your head, you'll immediately arrive at some qualitative, so not just quantitative, but major differences between what happens if this was like reaction diffusion, or if you think of it as well mixed, compared to if it's tethered. For example, if you run this, if you sort of in your brain think about what happens here and everything is untethered or just diffusing around, eventually you kill everything. The C's kill all the A's, or nearly all the A's. Maybe it happens exponentially or with a long tail, but eventually you deplete everything. But not when they're all tethered and immobile. Okay, and for me to see why I had to run a simulation, and the moment I show you the snapshot of the simulation, you'll see immediately why there's this difference. And then the other thing I should mention is that so this is the complex killing an A. This is when SHIP1 is in complex killing an A. It deletes it. This could happen. This also happens when SHIP1 is just in solution, but we expect this to happen much more rarely, because when it's in solution, it's effectively at a reduced concentration. Whatever it is, you still have this reaction. So this is our model. Really, there's just four parameters: K on, K off, Cat, and the length of this linker. Right, in the most naive version. Okay, so we want to model this. We think, how are we going to model this? We want sort of, for this particular problem, what is the best that we can do? And luckily for us, there's this separation of timescales that are occurring that says that the catalysis rate and basically all of the rates, so the K on and the K off, they are much slower than this other time scale, which is how fast the tethered molecule explores its little hemisphere. And in the language of polymer physics, this is called reptation time. So it very rapidly explores this little hemisphere that it's trapped in, and that's rapid compared to all the K's, K on, K off, K cat. And this means that we have the separation of time scales. Reactions are slower compared to reptation, and we can use this concept called effective concentration. Okay, that originates actually much earlier than this, and that says the following. Okay, if you have stuff that is not tethered, so for example, well mixed, and it's occurring at a macroscopic rate kcat times c, where c is the concentration, so that kcat times c is the per second rate, then if it's tethered, we can relate this to how fast the reaction will occur on a per second basis as k times sigma, where sigma is the probability density of this enzyme, this reactant, in its little cloud of constrained space. And in my language, and I think the language of most people here, this is what we would call a macroscopic rate. And so that means that we're coarse graining over the microscopic details of how these molecules are actually coming together. We just know that macroscopically, if they're in solution, you get some kind of thing that's proportional to concentration. And this makes sense, at least dimensionally. If you think about a probability density in 3D, it has to integrate to 1, and so it has units of per nanometers cubed, and that's micromolar. So this works out dimensionally. And the way it works out physically is if you have these two tethered molecules, you think about what is the distance between them, this r prime, and that has a probability distribution. The amount of time they spend close together will determine the reaction rate. And so if we assume that the tethers are worm-like chains or some other simple polymer model, then the probability density function is a Gaussian. And if you have two of them, then you have to sort of integrate this Gaussian against itself, and you get sigma. So I'll show you just what sigma looks like. So now this sigma, it's the probability density, but you can think of it as an effective concentration that will multiply the rates as a function of how far apart the anchors are. Okay, and so what we see is that if the anchors are very far apart, of course the effective concentration is reduced, because most of the time they're spending close to their anchors. And that's obvious, but the interesting thing is what happens when you change the tether length, L. And this sort of weird crossover region shows that there's a trade-off that makes sense in the following way. If the tether is very short, so like up here, then you just can't reach far distances, and so this drops off very quickly. So you don't want your tether to be too short. On the other hand, if your tether is too long, like this blue line right here, then you can reach far, but it's so dispersed in space that it gets lost and it can't find its substrate, and so that will also decrease your effective concentration and therefore decrease all of your rates. So we have this nonlinearity. The moment you have tethering, you have a nonlinearity in the tether length. Okay, so we can exploit that separation of time scales, and then we want to simulate this. And so this is our simulation. I think this is what most of us would think to do in this situation. We have the luxury of the particles being immobile. Okay, so what we do is we build a Gillespie time loop, and then outside of the time loop, we find the adjacency pairs, the adjacency matrix, and we collect all kinds of statistics. And it's n squared, but it can be slow because we do it only once outside of the time loop. And then once we know that, nothing ever changes. We know all the effective concentrations. Therefore, we know all the rates, and then we can just Gillespie iterate forward. Right, so what I'm showing here is in blue all of the phosphorylated layers that don't have ship 1 attached, and then they'll turn red when ship 1 attaches, and then it'll start killing all of its neighbors. Okay, and so this is what it starts as. It's all blue, but then it quickly becomes all red as the ship 1 attaches. But then ship 1 starts killing all of its neighbors, starts clearing space, and this continues for some time. We're decreasing the amount. Everything is dying. But then we get to some kind of steady state where ship 1 is attaching and detaching, but no one else is dying. No one else is getting dephosphorylated, for the obvious reason that you've now you've sort of cleared all the space that you can reach, and now everything is spaced to the point that you can no longer reach anybody, and so nothing else will become dephosphorylated. It'll just switch back and forth between phosphorylations. Right? So this is a qualitative difference between what would happen if it was well mixed. You just end up not getting to everybody in this case. Okay, yeah.
I got lost. You were flowing some solution over the substrate. So why is everything immobile now?
These are right. Good question. So what I'm showing here is only the anchors, and the blue anchors are when there's no ship 1 attached, and the red anchor is when there is a ship 1 attached. The ship 1 is still mobile when it's unattached, but the moment that it attaches, then it is immobile for the short
While
time.
this is happening, presumably some more could attach from.
Yeah, exactly. Yeah, and so that's why you get this equilibrium at the end where they're attaching, and there's new ones attaching. Yeah.
And the question: what's your tether length in this scale?
It's like tens of, on the order of 10 nanometers.
So you're 10 to what's the size of one of those red circles?
Yeah, exactly. Yeah. So that's why they're all cleared. Yeah.
What's the tether length compared to the distance between two points? Maybe this is probably important.
Between the distance between two particles. Yeah. Okay, so this is to scale. So at the beginning, it's very dense, meaning that the typical anchor points are much less than the tether length. But then at the end, because of the dynamics, it's just a little longer. So the tether cannot reach to the nearest neighbor. And that's when it stops killing people, because it can't reach them anymore.
So in your previous slide, you started with the separation of time scales, right? And you said that the diffusion coefficient for the tethered molecule is much larger. Since it's tethered, right, this only applies to short time scales. Long time scales, it's not going to move anywhere.
Yeah.
The diffusion coefficient is going to asymptotically go to 0.
Yeah. So this is you don't want to think about it as like so the root mean square would not increase linearly, right? So you want to think of this as the time scale for it to explore its confined region, and it's never going to leave that region. So it will have well explored that region on this time scale.
So as long as the reaction time scale is just a little shorter than this one and not much.
Longer.
Right?
As long as a little, it could be a little bit longer or a lot longer. Now we have the luxury that it's a lot longer. Yeah. So root mean square would not just keep increasing because it's confined. Okay, so that's our simulation. It's if you just do it naively, it's log n. There's nothing n squared about it once it starts running because we know where everything is. So we never have to compute pairs. Okay, so then we want to combine this with Ulmer's data, and so the data that comes out of these SPR experiments is you can see it's these black dots here, so it's extremely high quality data. There's really no random noise that you can see in sort of in fuzz in the data, and what we're showing is bound ship1 fraction, and just like in the simulation, it starts off at 0, then it increases as ship1 binds, but then it decreases as ship1 starts killing off all of its substrate, so then it can no longer bind. Then we do some fitting where we take the model without a priori values of the parameters. There's 4 parameters, remember, and then fit this all together, and then we get these parameters, these 4 parameters out. And this is, by the way, I'm sweeping under the rug on an entire effort that is necessary to make this fast enough that you can actually do fitting with it. That was sort of this heroic multipole expansion that Ulmer and one of his PhD students, Lally, did. Whatever this is, whatever I'm sweeping under the rug, you get these parameters at the end. And what you see is the k on the k off, you have very tight bounds. And so this is telling us that we're actually able to constrain all the parameters. In parameter space, 4 of them, right? So k on, k off, the length, which we estimate in the experiment, and then the 4th parameter was k cat, this catalytic rate. Okay. And so one of the things that we learned, I'll just remind you what the equation system is. It's yeah, there it is. There it is that really there's 2 k cats that a priori you might have thought were the same, and we thought were the same, but it turns out that we can only fit things if we separate them. Okay, so this is even after taking into account the fact that when you're tethered, the effective concentration is magnified by several orders of magnitude. That's still not enough to account for how slow the solution catalytic rate is by a factor of like 20 or something like that. And this teaches us something about the biology of this molecule, which is that when it's bound, it's active at a certain level, and then when it's not bound, it seems to be switching between states where it's less active on net. And this is consistent with other reports that use completely different techniques that have hypothesized that it has multiple states. Right? So already at this stage, Ulmer's learned something about the biology of this molecule. Okay, so then what we really want to do is understand the effect of the tethers, right? So the point of this is to understand why behaving on a tether is different than behavior not on a tether. And so Ulmer varies the tether length. This is the artificial linker. It's made of something called PEG, polyethylene glycol, and he varies it. PEG 28 is the longest, and you can go all the way to no PEG. And so it's shrinking from left to right. And so what we see is, so we redo the experiments, the fitting, all that stuff. And what we get is that when you estimate the L, that's the tether's reach length, this decreases as you shorten the tethers. Right? So this is exactly what you expect. We're shortening the length experimentally, and then when we fit, we end up predicting that the length is shorter, exactly as we expect. So that's good. But the bad news is that when we do this, we also find that the k cat and the k on also shrink as you shrink the tether length. Okay? And this is bad because if we had a biophysically predictive model, then you should be able to change the tether length. Only the tether length would change, and all the other biophysical parameters would remain fixed. And that's not what we're seeing here. So this tells us, so this is good in the sense that it tells us that we're missing some biophysics. There's something missing from our model. Okay, and the other rates are changed. So k off doesn't change that much. Okay, so around this, now we have to figure out what that is. And around this time, we started simulating at a finer length scale, but not that much finer. And so what we started doing is breaking down one SHIP1 molecule into domains And if you remember the cartoon from the beginning, the structural information about ship 1 is that it has 3 rigid domains. And so we're representing those as 3 spheres. 2 of them bind to a phosphorylated layer, and then one of them is the catalytic domain that actually does the dephosphorylation. And they're connected to each other by little linkers. And in this simulation, I'm showing it's bound to one layer, one tethered layer. The little link, the tether is shown in red here, separated by a distance little r to another tether that it's going to try to dephosphorylate. Okay, and so now we have it's coarse grain up from atomistic scales, but it's fine grain compared to what we were just doing, where we were treating ship 1 as just one particle, just one point particle. And so this is expensive to compute, but if we're just interested in equilibrium properties, we can use the Metropolis algorithm, which, so I'll just briefly describe it. It's all based on energies, and that energy can include steric repulsion, and you just generate new configurations, and you accept or reject them at some probability. So it looks like time dynamics, but it's not. There's no dynamic information. All it does is it gives you an ensemble that satisfies the equilibrium configuration ensemble of this molecule. So all it does is gives us equilibrium information, right? And everything just kind of jiggles around, and then you get these means. So we can get the effective concentration, because that's something that happens at equilibrium, especially on the slow time scales of our chemistry. So this is what it is when we use the mesoscale model. I'll remind you what it looked like when we just assumed a worm-like chain. And you have this weird crossover region. But when we include sterics and a little bit more detail about the molecule, that goes away. And instead it looks like this. So as it goes from blue to green, we're making the tether longer. And what you see is that at large distances, you're able to bind better with a longer tether. And that kind of intuitively has to be true. If you're far away, then you need the tether to be long in order to get out there. But we've lost this effect down at short lengths that we had in the worm-like chain case, where here now, at short lengths, if you make the tether longer, you can bind to you can hit your substrate better. Okay, and intuitively, the reason for that is because of all of the steric interactions with the membrane and with the other parts of the molecule. So if your tether is too short, then you're so close to this membrane that the things that you want to bind, the things that want to bind to you, just can't get in there, because the membrane's in the way or there's other molecules in the way. So you need a little bit of length before you can overcome steric effects. Yeah?
I'll ask later.
But in this version, you're also modeling the polymer, or like the flexible linker.
Yeah. Yeah. That's right. Yeah. Okay. So from this, we can get all this equilibrium information. If we just assume that, again, rates are proportional to concentrations, we know that this is the k-on that we predict. Now we predict it to decrease as we shrink our tether length. This is the k-cat. That should also decrease, because you've got to get the catalytic domain in there to have catalysis. And this broadly recapitulates the trend that you see in experiment. And also, one prediction of this is that because the phosphatase, the catalytic domain is larger than the binding domain, SH2, we would predict that this effect is stronger for k-cat than it is for k-on. And if you squint, you might be able to see that in the data. But I won't make that claim. Yeah?
How do you define effective concentration?
Okay, so the effective concentration is you have two molecules, they have a certain distance. That distance has a probability density. And that probability density's value when they're touching is the effective concentration. Okay, so it's exploring this space rapidly, and sometimes it's near, and when it's near, that's when you can have your reaction.
Is that two particular molecules, or the closest two that of the collections you're looking to react?
It's the closest two, but I should say that this simulation, there was only two. There were only two layers. Yeah. Okay, so this tells us that steric effects are one of the main determinants of our parameters, right? So there is actual crowding in this, or this is suggestive anyway, that there's crowding in our system, and now we got to include that into our Metropolis algorithm. Okay, so in other words, the picture does not look like this, what I showed you, where ship 1 is kind of like this point particle that attaches, but I don't know, maybe something more like this if we exaggerate a little bit. And it's crowding with each other, and it's also crowding with the membrane. The fact that we have this hard surface underneath it that it cannot penetrate is changing things, is determining the k-cat and the k-on. Okay, so how are we going to simulate this? This is what we want, and so just look at the picture on the right. We have these big spheres. Those are the bound ship 1. There's also, like I mentioned, there's unbound ship 1 that I'm just not showing here. That's like flowing on top of it. and they're all bouncing around. At some point, it'll get so crowded that they are behaving differently than they would if there were no crowding effect. Now, the size of ship 1 matters. And then eventually, if I played this movie long enough, they would start to decrease in total number because they've killed so much of their substrate. Right. Okay, so how do we do this? This is, I'll go through and the summary is really just on the right-hand side. We want to exploit this immobility of the anchors. So we do the same thing as outside the time loop. We get all the connectivity. We make an adjacency matrix. We get a lot of statistics, and it doesn't matter. It's order n squared, but we only do it once. And then we go to the loop. And in the loop, there is another loop where at every time step we do a metropolis sampling that does not give us any dynamics. We just move around the particles and get a configuration, an ensemble of configurations that allows us to compute the effective concentration. Then with this effective concentration, we feed that back to the Gillespie algorithm. Now we know all of our rates, and so we take a Gillespie step forward.
Yeah, I'm off. Sorry. I was getting nervous. So.
I told you that I would finish on time. I promise. Now I should go over just to spite you. Okay, so where was I? Right, so there's a metropolis loop inside the Gillespie loop. The metropolis loop is just about getting the equilibrium information that includes sterics. And then we do this Gillespie step. And the Gillespie step doesn't care where the particles are. All it cares about is where the anchors are, which doesn't change, and what the effective concentrations are. And that now changes with time as you get more crowded.
So now here it doesn't matter whether it's the nearest or the sum.
Yeah.
This is the sum of all big particles at B's that could touch an A. You add up all the concentrations.
Yeah, they're additive, but they're not independent because if you have a lot of crowding here,
That you add them up when you compare.
yeah. But you can shield other guys, and so they are competing with each other. Yeah, okay, so this is exactly what I showed you. On the left is the dynamics. This is Gillespie doing this, and then on the right is the equilibrium and the sterics is the metropolis doing this at every single time step. So this looks like time dynamics, but it's really not. It's just exploring a configuration space. And we just explore enough to get the concentration well estimated, and then we immediately go back to Gillespie, and then we take a step forward. And so what you're seeing here, these hemispheres are the domain ranges, and every little orange flash is when there's a dephosphorylation event. Okay, and by doing this, we're really doing next event, and for some reason the scaling is n with particle number. The reason I think that is, we're using a trick where we compute connected sets. And so every once in a while, if you look really carefully, you'll see one blue molecule that doesn't move around, that doesn't jiggle in the Gillespie. And that's because if it's disconnected from everything else that's happening, then we don't need to reevaluate it. And so I think our domains are big enough that even though the domain is getting larger, the active set is not, and because of that, we're observing sort of n scaling.
So just reassure me, if you wait for infinite time, eventually everything will go.
Not
But
every.
because you're flowing in at a fixed concentration and there's an underlying kcat solution.
Yeah. No. Well. Oh yeah. So there's a very slow kcat that will take care of things. But even if that wasn't there, yeah. So that will eventually kill everything. Yeah, that's right. So if that was not there, then you only kill so that you're sparse enough that you cannot reach your neighbors.
But you'd still theoretically kill because you have this Gaussian tail, so you have some non-zero rate.
Yeah, because of the solution. Yeah.
Well, not here. These are finite numbers.
Oh no. Yeah, that's right. These are finite tethers. Yeah. Yeah. Thanks. Okay. So now, according to my clock, you should stand up and scold me. For okay. So these are the results, and we're just working on this, but So these are two unmeasured variables: the amount of dephosphorylated stuff, phosphorylated stuff, and this is what you measure in SPR. So it's this characteristic increases, but then starts killing off its substrate, and the different classes of curves, or the different bunches, are different concentrations of SHIP1, and then the different colors are 1 nanometer in diameter, 2 nanometers in diameter, 3 nanometers in diameter, because its size matters now because there's steric effect, and what we see is it doesn't really make a difference. Okay, until we get up to around 10 nanometers, that's shown down here, where we start to see the rates sort of change in this kind of non-intuitive way. And if we can crank up the diameter of SHIP1 to something unreasonable, like 30 nanometers or 100 nanometers, right, so that's clearly larger than these signaling molecules. It's like the size of a vesicle or something. But I'm just showing this so that we can show our algorithm doesn't break down. It's still able to go through space, and we get these massive steric effects where basically nothing can bind because everything is being blocked by this this giant sort of beach ball. Okay, so that's it. We now have a simulation method that allows us to access this the sterics and reactions as long as they're tethered immobily and as long as there's a separation of timescales. Right? So this is a very narrow, I think, problem domain, meaning that we're not going to be able to take this method and apply it to more general systems, but within our framework, we're going to be able to extend it to understand what we need about the system. In terms of the biology, I think I'm experiencing what I think many of us are experiencing, judging by the talks, and many people in the field, if you look at the papers that are coming out, is that we're getting sucked into this scale where the interesting biology, the next frontier of biology, is sort of between where one particle is an atom or a residue or an amino acid and where one particle is a molecule, like a protein. Like if you think about all the simulations that I've done, one particle, there are at least 2 particles per molecule, per actual protein. And this is happening in my research all over the place. These are just 3 examples from my group. We have molecules being compressed at cell-cell interfaces, and to understand this, basically, we had to go to the scale where one molecule is not just a spring, let's say, but is actually extended a little bit. There's a long tradition of doing this kind of mesoscale modeling in the field of microtubule motors, kinesin and dynein family motors. We have, with a collaboration, we're studying this now, and we have a lot to learn, I think, from this field, because they've been doing this for a long time. And if you're the type of person who's interested in actin, we also have been studying this molecule called formin, one of the main actin nucleators. Basically, there's formin and up to 3, where by representing one formin molecule as this long chain, we're able to, we think, resolve some of the puzzling behaviors. So at this mesoscale that I think a lot of us are now starting to think at, from the point of view of abstraction, maybe none of this matters, because sort of in terms of the model at the end, it's just particles moving around and interacting. Or maybe it does matter, I don't know, but it certainly matters from the point of view of the modeler, meaning going from a scientific question and writing down what that model is at the first time. The challenges are different, the questions about how rigorous our model is are completely different, even if the computational tools will turn out to be similar or the same. Okay, like I said, Omer's been leading all of this research. There's people in his lab who've been working on it. Jesse is now a group leader at New South Wales, and Sam has been doing some of the simulation. And I'll just say I'm at UC Irvine Center for Complex Biological Systems, which is an NIH-funded National Center for Systems Biology, and we have a lot of exciting things happening there. So if you're interested in postdocs, visiting, or if you know graduate students, then you can tell them to look into this. Okay, thank you.
Thank you, and thanks for being on time.
Was I? I was, right? Yeah.
I think so. Okay, yes, please.
So just, I'm assuming that when you did the Metropolis, that was too expensive to then do the statistical fitting to see if that, then you can get constant rates independent of the tether lengths.
What would we fit to?
Well, I mean, somehow you need to show that this fixes the original problem, right?
Oh, that's right. Yeah, that's right. So we just haven't done that yet. Yeah, that's the direction that we want to.
Or it's just
Right now, it's too expensive to do. Yeah, yeah. So we, what I think we're going to end up doing first, at least, is not a fit, but rather to show that we get all the same trends. So it's like we're not going to explore a million different parameter sets. We're going to do a few and show that we can sort of get the ballpark. Yeah, Omer, you want to add?
Well, I just want to ask a question. So does that mean, in that case, would you have another parameter, like the size, the effective size of ship one?
Yeah. You introduce one new parameter. So now you have, instead of 4 parameters, you have 5. Or 6 instead of 5.
So in the end, is the tether stretchable or fixed?
So we're modeling it, following some, I think, fairly conclusive experimental evidence on other tethers as a worm-like chain, which is not, once it's completely straight, it's not extensible. But these polymers tend to be really bunched up, and so you can think of it as like a spring. But really, it's because it's unfurling, not because it's actually stretching once you pull it past its length.
So question about the tethers. So you have a tethering density on the substrate. It's a question of if you can experimentally vary it, but also do you know that you link at the end and that the tethers are all monodisperse in length? So there's questions about how you're putting the tethering into the model, and how well is that characterized experimentally?
So this might be for Omer, but the tether length I'm pretty sure is fixed because I think you buy them as peg 28s, and so they're all just peg 28s. If you can trust what the company says. And the second one, and then we know at the end that it's attached exactly at the end. And then at the surface part, do we know that it's attached at the very end of the peg? Yeah. Okay.
And so you're not putting in sort of polymer brush effects. So your tethers are at a low enough surface density that you can ignore that, or?
Yeah, for now. Although if we want to get to the densities that we see in perhaps that are more relevant for clusters, then it'll become a polymer brush, and we'll have to put that in. Yeah.
Kind of following that up, is the density, is the configuration of the tethers on your model like on a square lattice or something, and how is that captures the experiment? Is there any information what's the distribution of like maybe there's some sort of interesting kind of length, like spacing distribution that might affect the results and something?
You mean like one individual tether?
Distribution of tether points on the surface. Is it just
Oh, no. So we model them not as square lattice, but as 2D Poisson. So they're random on the two-dimensional surface. And how accurate is it? How much does that correspond to experiment? I don't know. Maybe Omar can answer that. We just assume that they're random, right?
Yeah, I think that's reasonable. We could vary them. We could make them more clustered, I think.
More clustered.
I have a question. So what are the strongest stochastic effects that you are looking for, sort of, that you think are there somehow?
Stochastic compared to the mean. So one thing is that we can't stochastic compared to like well mixed. It's a strong effect because one tether doesn't see everybody like it would be in a well mixed system. One tether only sees its neighbors, which is a very small number. It's like
Could one think of deterministic models that still incorporate this finite?
Yeah. In fact, there's this multipole expansion that Omer's student has been looking at, where it's just a series of integral PDEs. Yeah.
More questions? Yes.
From this, I don't see. How do you answer the questions why you have receptor clustering, for example, from this analysis?
Yeah. So we're still quite a bit of a way from there. But what we're going to get from all of this, one of the things it tells us is just how much that enhancement of concentration is playing a role. Once they're clustered, all the reactions are magnified because everything is just the effective concentrations are so large, and so everything will happen faster. And then we're going to look for other effects, the interesting things that might happen when you're in a cluster compared to not. For example, because there's crowding, there's not crowding when everything is just when everything is monomeric. So when there's crowding, you could get some effect where you expect some quantity to increase, and instead it decreases. But we just don't know yet.
But the receptors still move. There's not crowding that they are stuck in the membrane. They still have a diffusion coefficient. So it's not that like here they are tethered somehow. Their mobility is reduced, right? And right now this setup is the limit where the mobility is down to 0. But the mobility will be reduced when you have this tightly packed cluster. The guy in the middle is not going to be able to. Yeah, when they are clustered. But before they are clustered, they are.
Yeah. So this is not applicable to before they're clustered. Yeah.
I seem to remember there's a Birkhoff technique where you do have mobile receptors.
Yeah, I think you can, and I think people could build this with mobile, right? And I think we talked about doing that.
There's a chip that has a planar bilayer.
Yeah.
Yes.
Probably not related, but the creation of the eye by clustering of photoreceptors allows you to analyze more complex things, but I'm not sure if the nature of signaling is the same.
Okay, thank you. Yeah.
Okay, I think that's it. Let's thank the speaker once more.
Thank you.